Continuing with blood, we're going to talk about the formed elements. Erythrocytes are red blood cells. They're the most common element in the blood. It's what gives blood its essential character. These are basically specialized cells that have a function of transporting gases. The gases that they transport really are oxygen, which binds to the hemoglobin, and a little bit of carbon dioxide, which also binds to the hemoglobin. Within the red blood cell is also where it's catalyzed that the carbon dioxide becomes carbonic acid, which then becomes bicarbonate ions. And so it's involved with the creation of bicarbonate ions for carbon dioxide transport. But mostly it's just basically taxis that carry oxygen through the body. And so when we have problems with erythrocytes, it manifests as problems with oxygen delivery. Now these cells are what are called biconcave discs. Basically what they are little spheres that are pinched in the center. They look just like the snow tubes at Snow Valley, and the reason they're like that is for the same reason that the snow tubes at Snow Valley are like that. It makes them really slippery, easy to move, they don't tangle with each other, they bounce off one another, they move down the pipe very quickly, the same as the snow tube. The other reason they're like that is that what it means is that the surface of the cell ends up going right to the middle, or close to the middle, so it enhances the diffusion capabilities, and so the oxygen exchange. Oxygen can get to all the hemoglobin, not just the hemoglobin that's just below the surface. So that's an important thing. Mature erythrocytes have no nucleus. They have no need for a nucleus. Once the immature ones do have a nucleus, but once messenger RNA is transcribed for hemoglobin and spectrin and some of the other proteins that are needed, mostly it's hemoglobin, then there's no more need for the nucleus, and the nucleus just ends up taking up space that could be used for hemoglobin to transport oxygen, and creates weight that makes it more difficult to move. If we go back to the last video for a second and think that there are 5 liters of blood in the body on average, and the average at-rest heart rate pumps 5 liters of blood per minute, that means that all the blood in your body has to go everywhere, through every capillary in your body, in less than a minute. It has to be transported, passed through the capillaries, and then back to the heart and through the lungs and the capillaries of the lungs, and then back to the heart, all in under a minute. So weight is a consideration. They don't have a lot of organelles either, because, as you know, especially mitochondria, there are no mitochondria in erythrocytes, because really you don't use a dog to deliver meat. If you mitochondria use up oxygen, and if what this is a transport system for oxygen, let's not have it consume the oxygen that it's transporting. There's so many of them that they really are the major contributor to blood viscosity. It's the reason that blood is thick and somewhat sticky, is the red blood cell. When you look at it, it looks like this, the healthy ones do, and you'll see that it's 7.5 microns. What's by 2.5. It's interesting because the average capillary is only about 8 micrometers in diameter, the lumen. So that means that in the capillary beds, these things, these red blood cells, the billions of them, have to go through in single file. And there are some capillaries in the spleen that it's actually about 7 those capillaries are a little bit smaller, and each one of these red blood cells has to go through folded because it can't fit through unfolded. These guys are very bouncy, kind of rubbery sorts of things, and so they will deform, which also helps them not, healthy ones, not clump together, and they stay apart. So the biconcave shape is it makes more surface area to volume. It's mostly hemoglobin, no mitochondria. So any energy that they produce is anaerobic. They are completely functional things. Now, a byproduct of this is that they're unable to repair themselves, and any ATP that they need contribute to an acidity. So these cells actually wear out. They don't reproduce because there's no nucleus, so they can't undergo mitosis. So really, you make them, you use them. When they get old, you get rid of them. We already talked about this. Respiratory gases, oxygen being the most important. I'm not so worried about the structure of the hemoglobin. It doesn't matter that there are 2 alpha and beta chains. What matters is that there are these pigments, the things that actually contain the iron, called hemes. And the globin part is just a protein, a structural protein. In the center of each heme is an iron molecule. So when we look at it, it looks like this, and it doesn't matter what the pigment, what the heme pigment actually looks like. The salient thing is there's an iron molecule in each one, and each hemoglobin molecule actually has 4 of these hemes. And it's the iron in it that binds with the oxygen. So each hemoglobin molecule will carry 4 oxygen molecules. We'll see later in the destruction, in the life cycle of the red blood cell, that we get to reuse all the amino acids in the alpha and beta globin chains. We get to reuse the iron in the heme, but we don't get to reuse the rest of the heme. So it becomes waste material. In class, we talked about hemoglobin loading and unloading, and oxyhemoglobin and reduced hemoglobin, and carbon dioxide loading. And carbon dioxide, 20% of it is attached to hemoglobin. So where do these things come from? We actually call any blood cell production hematopoiesis or hemopoiesis. Either name is correct and acceptable. One it tends to be more American, the other one tends to be more British, but it really means blood formation process. It happens in the red bone marrow, which is found in the spongy bone, the cancellous bone, usually at the proximal ends of long bones, and so it's in your pelvis, in the proximal femur, and the proximal humerus is where most of it happens. There's a little bit of it other places, but really it's hips and shoulders is where most of this happens. There are stem cells that live in the bone marrow, and they're called hemocytoblasts or hematopoietic stem cells, and they really are the precursor cell to all the blood cells, all the formed elements. And depending on the body's needs, these stem cells differentiate into the different types of formed elements. The hemopoietic stem cells undergo mitosis and are constantly reproducing themselves. And understanding this really leads us to understanding things about leukemia and such, which is really a problem of these cells. Like, it's a cancerous alteration in these stem cells. These blood cells arise outside of the blood vessels, and they enter the blood capillaries when they're ready, when they're mature, through special capillaries that have large holes in them, and those capillaries are called sinusoidal capillaries. A sinus is a large hole, so sinusoid, so it looks like a sinus in the capillaries. Red blood cells obviously have to be made at a prodigious rate. We make lots of them. We have billions of them. We are losing thousands, if not millions of them every minute. We have to produce them at this rate. So what happens is this. Sometimes they're called pluripotent stem cells, which means that they can become anything. The hemocytoblast, the hematopoietic stem cell, gets stimulated by a hormone called erythropoietin that makes them transform into something called a proerythroblast. Now, I'm not going to expect you to know all the stages of this, but basically what happens here is that because of hormones, it starts on a course of changing. So the original stem cell, the hemocytoblast, is really a nucleus that has all kinds of genetic information, and it is going to become, it has the potential to become other things. The cytoplasm is basically for support and nourishment of the nucleus. Under a certain hormone, what happens is the nucleus kind of gets turned on, and it starts making ribosomes in the nucleolus. It starts to transcribe the correct genes for making hemoglobin and spectrum and some of the other proteins that are necessary. But basically, at this committed cell stage, the proerythroblast, what's happening is this is when the mechanisms are getting turned on. If you're going to build Hondas in Alliston, the first thing they had to do was decide where the plant was going to go. Then they had to build a plant. Then they had to build all the assembly lines in the plant before they built their first car. It's the same thing. The stem cell is the open field. The committed cell is the planning of where the plant's going to go. Then in the early erythroblast, you basically are building the plant. You're building the organelles that will assemble the proteins. In the late erythroblast, you've made all these ribosomes, and you're starting the process of translating them into proteins. You're bringing on a bunch of amino acids, and you start making the hemoglobin proteins. A little bit later, as the hemoglobin starts to accumulate in the cytoplasm, has got way more hemoglobin than not. It's called a normoblast, and during all of this, these ribosomes are on endoplasmic reticulum. At about this point, there's no sense in having the nucleus anymore, because you've made all the messenger RNA that you're going to need, you've made all the ribosomes you're going to need, you've made all the endoplasmic reticulums you're going to need. So what you end up doing is saying, well, this nucleus is just taking up space and not doing us any good. So the cell really just ejects the nucleus by exocytosis. It takes the nucleus and spits it out. And all that's really left then is endoplasmic reticulum with ribosomes on it, and it's accumulating all kinds of hemoglobin because the hemoglobin is being made on this endoplasmic reticulum. Once the nucleus is gone, we call it a reticulocyte because it's just endoplasmic reticulum and hemoglobin. This, at the reticulocyte stage, is when it gets put into the bloodstream, and so we can look at blood, and the more reticulocytes we see, the more we know that red blood cell production is up, because we see these immature things. They can carry oxygen, but they're not as efficient as the fully mature erythrocyte. As the thing absolutely fills up with hemoglobin, then we call it a erythrocyte, and it takes on this typical biconcave shell. So the phases are: first you make the protein synthesis machinery, the ribosomes and the endoplasmic reticulum. Then you start to make the hemoglobin. Then you get rid of the nucleus and become reticulocytes. Then turn into full-on erythrocytes. If you don't have enough red blood cells, then you're not going to carry enough oxygen. You might be breathing enough, but you're not going to deliver the oxygen that the tissues need. So the tissue ends up having hypoxia, and you get symptoms of hypoxia, which really fundamentally is you're tired because you don't have the oxygen to make the ATP. If you have too many red blood cells, then the blood becomes too viscous. It increases resistance to flow, and it makes blood flow more difficult. So you have to have just the right number of red blood cells. And so really, anemia is too few, and hypercythemia is too many. So you got to keep it just right, and we do that hormonally, and we also need the raw materials to come. So you need lots of iron, you need lots of amino acids, you can imagine, to make all the hemoglobin, and you need B vitamins. Those are important as well. The main hormone that controls erythropoiesis is called erythropoietin, EPO. It directly stimulates erythropoiesis in the bone marrow. Now it's interesting, the organ that causes the release of EPO is the kidney. And the kidney actually uses a lot of oxygen. It's the number 3 user behind the brain and the heart of oxygen. So it's specially attuned to hypoxia. Like it will suffer from hypoxia before other organs because it uses so much ATP. It runs out of ATP quickly, and in response to that, releases this erythropoietin. One of the interesting things about this is that that means that anybody who has kidney disease is probably going to be suffering from an anemia, probably not going to make enough red blood cells. So if you ever work in a dialysis unit, one of the things you're going to be doing is giving people erythropoietin because they need it in order to avoid anemia, because their kidneys aren't producing it properly. Almost invariably, somebody that's in kidney failure needs erythropoietin. Because of that, there's this synthetic EPO, and it can get abused by athletes to try and raise their hematocrit to raise their endurance. Very hard for them to find it because erythropoietin is a normal thing in the body. Erythropoietin gets released by anything that can cause hypoxia getting to the kidneys. So that could be a loss of red blood cells. So anything that reduces the red blood cells number, like for instance, bleeding, hemorrhaging, will reduce, and so it will stimulate erythropoietin, and so that we get more. If red blood cells are being destroyed by a disease process, an anemia, it will cause hypoxia, which will cause erythropoietin to be released. If you don't have enough iron, you can't make functional hemoglobin. If you can't make functional hemoglobin, then it's going to lead to hypoxia. If there's a reduced availability of oxygen, it's going to happen too. So one of the things that happens is people, like say people that have COPD and aren't getting enough oxygen, release a lot of erythropoietin so that they can have more red blood cells to carry the oxygen that they do get to the tissues, which works for a while, but then that means that their blood is sluggish, that it needs to be forced more because of the viscosity, so they often undergo blood pressure issues because of the increased number of red blood cells, and it can lead to strokes and blood clots and other nasty things like that. So people that have this chronic hypoxia have this problem. When you live in Denver, there is less oxygen available because of pressure things, not because there's less oxygen in the mix. It's just there's a lower atmospheric pressure, so the partial pressure of oxygen is down. So anytime you're at altitude, you will have a reduced amount of O2, which will lead to hypoxia, which will lead to erythropoietin, which leads to an increased number of red blood cells. And that's why a lot of athletes actually go to Denver to train and then try and compete at sea level, because it actually increases their oxygen delivery capabilities. Testosterone enhances EPO, and one of the things then is that men have more erythropoietin and have then therefore a more hematocrit than women. So it looks like this. We have hypoxia, so low oxygen in the blood. You could have decreased red blood count, you can have decreased availability of oxygen or decreased hemoglobin, and it causes the kidneys to release erythropoietin in response to the lack of oxygen. The erythropoietin stimulates the red bone marrow and commits the stem cells to becoming erythrocytes. A few days later, the red blood cell count goes up, which means that you carry more oxygen and get out of the hypoxic state. That's the theory, and in normal healthy people, that's what happens. You need lots of amino acids, you need fats for the cellular membranes, you need lots of carbohydrates, you need iron, and you need B12 folic acid in order to make this happen. So people that are low in B12 end up somewhat anemic and they're tired. Same with folic acid. The same with not enough iron. Now, most of the iron is found in the hemoglobin in your body, but we have stores of iron in the liver and in the spleen and some in the bone marrow. It gets stored as ferritin or hemosiderin if it's found in the blood. When it's moving between say the liver and the bone marrow, or the spleen and the bone marrow, or the spleen and the liver, it attaches to a protein, and it's called transferrin because that transfers the iron. Ionic iron is very toxic, so we have to transport it bound. So these guys live about 100, 120 days. So, it's someplace around 3 or 4 months. Literally, you don't have any of the same red blood cells you had when you started this program in September. What happens is, because the red blood cells can't fix themselves, they become fragile. The hemoglobin itself starts to degenerate, the proteins, the other proteins start to degenerate, the cell membrane starts to degenerate, and the whole thing ends up just becoming fragile and become problems. So what happens is they go into the spleen. 20% of your blood passes through your spleen every circulation. I believe it's about 20. But nevertheless, within 5 or 10 minutes, all your blood passes through your spleen at some point. Now, the capillaries in the spleen are smaller than the red blood cells, so the red blood cells have to fold to get through the spleen. If they can't fold, they get stuck at the opening to the capillary, where there are resident macrophages that just eat them and digest them and recycle the bits. So when the macrophages eat them, the heme and the globin are separated. The heme is then separated, and the iron is salvaged to use. And then what's left is this yellow pigment called bilirubin. Bilirubin. So rubin means red, red blood cell in the bile. So what happens is then that ends up getting pushed over to the liver, where the liver mixes this bilirubin as an excretion product into the bile, which then gets squirted out into the intestines, and we get rid of it. Now, it's yellow in color. When it gets put into the intestines, the pigment starts to degrade, and it ends up turning brown, and that's why your poop is brown. Some of this bilirubin ends up being excreted in your kidneys if it doesn't make it to the liver, and that's why your urine is yellow. It's old red blood cells. It's the pigment of the old red blood cells. If your liver is not functioning properly, then this bilirubin starts to build up in your tissues, and that's where jaundice comes from. So this just becomes kind of a chart outlining all of this. So when we get to the spleen, the hemoglobin gets broken into the heme and the globin turns into amino acids, which are secreted back in and used elsewhere. The heme gets broken into bilirubin, which goes to the liver and is secreted out as bile. The iron gets made into transferrin, which then is sent to the liver and stored as ferritin or hemosiderin, and then it gets transported as needed from the liver back to the bone marrow, where you use it for the next erythropoiesis. So, low oxygen, erythropoietin levels rise, new erythrocytes enter the bloodstream, and they last for about 120 days, and then this is what I just talked about.